Good day, Mindsetters. My name is Megan, and welcome grade 10 to today's Life Sciences show. Today we have a special revision lesson on the chemistry of life, prepared especially for you. We'll be selecting highlights from the lessons shown early in the term to help you revise for your end of term tests. You can download the notes for today's show on learnextra.co.za/live.
Guys, we're going to have awesome time this year. The best time to tune in is after school on a Wednesday afternoon when we do life sciences. So we've got the grade 10 lessons in the afternoon from 4 to 5, and then we have grade 11s and 12. But for you guys that are in grade 10, you've got to remember that this is your first year of life sciences. And essentially we try and do, during this first session, we try and do what we call the chemistry of life, which forms the basis of biology, or life sciences, so to call it. So we're going to spend lots of time today doing understanding what chemistry of life is, understanding what organic and inorganic molecules are, so that when we go on to the next chapters, when we talk of cells, we understand what cells are made up of. When we get into grade 12, when we talk of DNA and RNA, we understand exactly what DNA is. Or if we talk of carbohydrates, we should know what carbohydrates. So essentially this is very important. So you're going to spend lots of time doing this. We're going to go through today, we're going to discuss the organic compounds as well as the inorganic compounds before we get on to doing some nice food tests next week. So next week, Katleka, we're going to do lots of food tests. We're going to do lots of practice. So you guys must tune in. And so we're going to have lots of fun next week. So let's get on to what we've got to do for today. So we're looking at chemistry of life. And often when guys hear chemistry, or when you guys hear chemistry, go, oh no, this is going to be a chemistry lesson. No, guys, this is going to be about the chemistry of life of biological modules. So we're going to look at not the details of chemistry, but understanding the chemistry of molecules in terms of when we talk of a protein, whether it's an inorganic molecule or an organic molecule. So let's look at what we have today. Right, so during this lesson, we're going to investigate the structures and functions of molecules that are essential for life. So today we're going to look at the essential molecules of life. During this lesson, we're going to look at the terminology, and that important terminology is we often come across words such as molecules, and we come across words such as atoms. So let's look at what an atom is, or what an element is. So today, a molecule is a chemical structure made up of atoms. So when we look at atoms, it's basically several atoms put together that give us a molecule. We often hear of the word a compound. And basically a compound is a chemical substance that is made up of two or more elements. Now guys, during your grade 9 year, you looked at the periodic table and you looked at the various elements. During this chapter, we're going to look at how those elements are put together to give you compounds. And essentially, if you think about water, H2O. H2O is made up of elements. So we're looking at how those elements come together to give you biological compounds. And that's essentially what we're looking at today. So here we have a diagram that shows you the relationship between elements and atoms, compounds and molecules. Now, if I were to ask you this and you were in my class, what would be the bigger between these two? What would be a larger molecule? And we know that an atom is obviously very small. So we learnt about the structure of an atom in grade 9, and we know that atoms are much smaller. And atoms make up elements. So we see that there's a relationship between atoms put together forming elements. We also know that atoms make up molecules, and we get molecules that are made up of atoms. What we want to see today is the relationship between a molecule and a biological compound. So by that we refer to looking at simple molecules being put together to give you complex compounds or simple compounds. And that's essentially what we're going to discuss during today's lesson. Okay, so when we look at molecules in a human body or in a plant, we can classify them into two main groups. We can look at compounds as organic or as inorganic. Now, we often hear of the term organic substances. Now, essentially, how would you differentiate between an organic substance and an inorganic substance? Now, I challenged my learners, and so they came up with definitions like they contain carbon and they contain hydrogen, or they're held together with hydrogen bonds. So let's look at that, because we also have some inorganic compounds that contain hydrogen and even contain carbon. So we cannot use that statement to say that all organic compounds contain only carbon and that inorganic compounds do not contain carbon, because there are inorganic compounds that do contain carbon. Let's look at how we unpack the difference between inorganic compounds and organic compounds. So essentially, an inorganic compound, these are compounds made by a natural geophysical process. So they're naturally made. They're not produced through a biological process. They're naturally made in the environment. They form in living organisms as part of metabolism. So they could be formed in a living organism as part of a metabolic process, and that's accepted. So common examples of inorganic compounds are water, carbon dioxide, oxygen, sulfur dioxide, as well as sodium chloride, and the list can go on. Essentially, if we look at these inorganic compounds, we see that they have hydrogen, they have oxygen, they have carbon, and they have the other elements. When we look at organic compounds and what differentiates them from inorganic compounds, we would see that there's the presence of carbon, hydrogen, and oxygen, and some of the other essential elements. So let's look at the definition of an organic compound. So an organic compound is a compound that is made through a chemical reaction in a living organisms. And that's very important, guys. Organic molecules are produced through a biological process. So if we think of biological processes, we refer to photosynthesis and we refer to respiration. We know that plants carry out photosynthesis. That is a biological process. And they have the ability to take the elements, so we talk of water and we talk of carbon dioxide, and put that into a biological compound. And by that we refer to that as a physiological process. So the carbohydrates or the sugars made by plants are an example of organic molecules. And essentially we say they're organic because they've been produced through a chemical process in a living organism. Okay. They are nutrients and energy sources. If you look at the bulk of our food, and all our food actually, they are organic compounds. Essentially they are made through a biological process. Either it's made in a plant or it's made in an animal, and we consume these organic molecules. And they provide us with energy and they provide us with nutrition. And that's essentially how a cell will survive, or our body survive. And that's why we refer to this chapter as the chemistry of life, understanding and unpacking the chemistry behind the molecules that are important for life. They must contain carbon, hydrogen, oxygen. Some may contain nitrogen, sulfur, and phosphorus. So when we look at our organic compounds, we see that they contain carbon, as I said here, hydrogen, oxygen, nitrogen, and sometimes sulfur and phosphorus. So essentially, an organic compound will contain carbon, hydrogen, oxygen, and the other elements, as opposed to an inorganic compound, which is not made through a biological process, as it is made through a geophysical process or a process not biological, and hence that would differentiate the two. Let's look at some examples of organic compounds that you will look at during this year. So we often hear of the word carbohydrates, okay, or we often hear of people talking of a high carb diet or a low carb diet. Proteins, we know proteins are very important in our diet. We talk of fats, we talk of nucleic acids, and we're going to look at vitamins as well. So these 5 compounds are your organic compounds, and these organic compounds form the basis of living organisms. They're important for nutrition, they're important to sustain human and animal life. So let's go on to we're going to look at the inorganic compounds today, and the first compound we're going to look at is water. And we know that water is essential for life, and when you look at the structure of water, guys, water is an H2O molecule compound, and it has 2 hydrogen atoms and an oxygen atom. If we were to draw a simple structure of a water molecule, you'll find that it has 2 hydrogen atoms, so that's 2 hydrogen atoms and an oxygen molecule, H2O. So let's look at the role of water or its importance. We said that water makes up 70% of our body, and we know that what is essentially important for us, we don't have water, we dehydrate. Water is also the main component of our blood and body fluids. A large percentage of blood is actually water, and the body fluid that we have in our body surrounding the cells and tissue is essentially made up of water. So if we talk of tissue fluid, the composition of tissue fluid is water. So water plays a very important role in our bodies. Water is also important in dissolving other chemicals in our body, and it allows for chemical reactions to take place. So we refer to water as a universal solvent in which other substances can dissolve or other solutes can dissolve. So in our body, water forms the basis of all chemical reactions. So it's a medium in which chemical reactions take place. So if we don't have water, we will not be able to break down substances, neither will we be able to build up substances. So water plays a very important role in the chemical reactions in our body, and we refer to those chemical reactions as metabolism. Okay. Water also plays an important role in removing the waste products from our body. So urea, sweat are all made through water. So you have urea, uric acid dissolved in water, you have your waste products that all are passed out through water in our body. So when we excrete waste, lots of the waste contains water, and that's how the body flushes out the excess waste in our body. When we exhale carbon dioxide and water vapor. That's also water produced as a result of exhalation or respiration in our body, and that is to get rid of the excess carbon dioxide and to maintain the body constant or homeostasis. Water helps to regulate our body temperature. And guys, it's summer now. You know that we sweat a lot, Katleko, and you notice that often when you're driving in a car and you don't have your windows, you don't have an aircon on, but when you do put those windows down, you find that the wind actually cools you down. And that's because the sweat on your surface of the body are water droplets that actually cool down.
Okay.
So what happens is that your body cools down because of the sweat, the water droplets cooling down. So it may not play a significant role in us, but you'll find that in animals, this is an important mechanism of cooling and maintaining their body temperatures. So water plays a very important role in cooling the body and maintaining the correct temperature. Water is lost through urine, sweat, and must be replaced. And we know that we need to rehydrate ourselves, drink lots of water, and that is to maintain the body volume, the blood volume in our body, because if we don't, we dehydrate, and that's not good.
How many glasses of water should we be drinking?
Well, people say that we need to drink 4 liters of water a day. I think it's important to drink water all the time, and essentially to keep yourself hydrated. The body has a thirst mechanism, and you find that when you're thirsty, the body tells you that you need to drink water.
Yes.
Unfortunately, that is not a good practice. You need to drink water more often than when you feel thirsty, because feeling thirsty is a mechanism in the body to tell you that your body is lacking water, and that's not ideal. You should be drinking water not when you're thirsty, on a constant basis.
So your body should never lack water.
Yeah. The thirst sensation is the body telling you that your body is lacking water.
Okay.
Okay, and you'll notice that you feel less energetic when you're tired, and often a glass of water or two helps you re-energize. It reconstitutes your body blood volume, and that's important for you. Okay, so we're almost there for a break, Katleko, but I'm going to in a very quick nutshell, I'm going to go through the importance of water before we go in for a break. So we know that it is a universal solvent, guys. It is a transportation medium. You'll find that in plants and humans. In plants, water is needed to transport the nutrients from the soil to the leaves, and that's for photosynthesis. In humans, as we digest food and that's absorbed into the blood, it's transported to all parts of our body. It also forms a medium for chemical reactions, and we know that reactions in the body will not take place without water. In plants, guys, water gives plants shape. So plants do not have a skeletal system, and you'd see in a plant in summer, as soon as it gets by the afternoon, they start drooping, hanging, and that's because they've lost lots of water through transpiration. So plants rely on water to give them shape and turgidity, and turgidity is basically referring to being a cell being firm and having proper shape. And most important, water plays a very important role in the removal of waste products from our body. So we've looked at, in this session, we've looked at inorganic compounds. We've looked at the importance of one of the most important inorganic compounds, which is water.
Let's go to a short break now. Remember, if you are struggling and you need help, remember to post your comments and questions on our Facebook page, facebook.com/LearnExtra, or on Twitter @LearnExtra. Don't go away. We'll be right back. Welcome back, guys. Let's get into some more revision.
Good afternoon, guys. As promised last week, we're going to look at some of the experiments on organic compounds. Just a brief chat about what we did last week. Last week we looked at the inorganic compounds, and then we discussed a bit about what the inorganic compounds were, and I said it's important to remember the source and the abnormalities and deficiency symptoms that arise from a deficiency in an inorganic compound. Today we're looking at the organic compounds, but we're looking at specifically testing for the presence of organic groups in different food samples. So what we have here today is, and we're probably going to do about 4 experiments by the end of the session, so we're going to have a power-packed session of 4 experiments where you're going to look at different food samples and you're going to test them. Just to very quickly touch back, we discussed the 4 groups last week. We said they're proteins, they're lipids, they're carbohydrates, and obviously we looked at proteins. Today we're going to be testing for proteins, we're going to be testing for the presence of lipids in different samples. We're also going to be doing a starch test to test the presence of starch, as well as we're going to do the glucose test to test the presence of glucose. So what I've set out today here in front of you is I've got different food samples that I've prepared. And here you see that there's a glucose sample. Here we've got a starch solution. I've got some grape juice in there. I've got some milk that we normally have with our coffee and tea. We have got a wine sample that we use for cooking. And here I have an egg solution obviously prepared from there. Now, at the start, I'm not going to actually mention to you what these would contain. But we know that they are proteins, they are carbohydrates, and they are fats in these different food samples. We also have, later on in the episode, we're going to look at the nutritional value of having peanuts and different types of nuts and why they're important to us. So let's start. Alright, so today the first test we're going to look at is for the testing of glucose. And guys, when we test for the presence of organic compounds, we need to have reagent. Reagent basically is an indicator. It's a mixture of a few chemicals that will react with the chemical group and it will produce a color. And essentially what we're looking for is we're testing for the presence of a protein or an organic compound. That could be indicated by the presence of by a change in color or by the absence of a typical color. So when we look at a food test for glucose, we normally test for the presence of glucose using a reagent known as Benedict's reagent. Now guys, you probably would have seen other reagents that you work with, but we tend to use lots of Benedict reagent. It's easy to use, it's very quick, and we get results very quickly. You would need to have some boiling water, and remember guys, whenever you do an experiment, you've got to take the necessary precautions. You don't smell any of the contents, you don't sniff out any of the contents, you don't taste anything that you have, and that's important. And ensure that your windows and your rooms are well ventilated. And always guys, do an experiment with your teachers around, so that if something goes wrong, your teachers know exactly how to correct it or how to react in the situation. So let's start. So what I have here is I've a set of test tubes and I've labeled them glucose, I've labeled them water, grape juice, milk, and this is egg solution, and here we have the starch solution. So what we're testing for in the first test, guys, is the presence of glucose in different food samples. And if you notice, the first test tube is a glucose test tube, the second one is a water. And we call these the positive control and the negative control. And the reason why we have these is because in order for us to know what the color change is of a substance that contains glucose, we would need to have glucose in that series of experiments. So we have a positive control as well as we have a negative control, which is water. So we're going to throw in 2 ml guys into the first test tube, and it's glucose that goes into the first test tube. We're then going to add negative control, which is water. We're adding a similar amount, and we know that water does not have any of the carbohydrates in there. It's an inorganic molecule, and hence this is our positive control, and that's our negative control. And so this would be used to compare results against the others. The next tube is labeled grape juice, and so we're going to throw in some grape juice in there. And we know that grape juice is sweet, so we're expecting some results here, guys. So we're going to put in some of the 2 ml of grape juice into that test tube, and then we're going to add in some milk, guys. And we know what the value of the nutritional compounds in milk is, but we're not going to guess yet. And let's see if we get results that we anticipate. So that's one that we put in there. And the next one is an egg solution, guys. And we know that eggs are very important. They're important as a source of protein, and you know why that's in your diet. So it's very important to sustain your protein needs by having lots of eggs. And obviously the last one is a starch solution, guys. And we know the value of starch as well. We see that coming from your potatoes that you eat. Let's look at so we've created now a series of 6 test tubes. And the important thing is to make sure that you've got your test tubes labeled. We then going to add in an amount of Benedict's reagent, which is what we're going to add. We're going to try and add in about 2 ml of Benedict's reagent into each one of these. Okay, that's Benedict's reagent to the first one, to the second one. And while I do this, guys, I've got a kettle that's boiling, and that's obviously I'm going to put these test tubes into the kettle and allow them to stand for about 15 minutes. And while that happens, we're going to start with the next set of experiments where we're testing for the presence of proteins in different food samples. So now we've added Benedict's almost 12 over to each one. We hope that nothing's going to blow up today. But it does, because we're using organic compounds and we're not using anything that's combustible yet. Because we're getting ether, and that's really give us a gentle stir, and we're going to look out for a color change. Now, the color indicator we're using Benedict's reagent guys is a dark orange-red precipitate that will develop at the bottom of the test tube, and that will indicate a positive change. But these test tubes will need to go to water bath. And there's a reason why that they go to water bath, and that's important to understand. Remember that the reactions are speeded up when we provide heat. And to speed up the rate of the reaction so that and provide the energy for the reaction to take place, we're going to immerse these test tubes into a beaker filled with hot water. So I'm going to quickly grab a kettle, fill it up, and I'm going to put in these beakers into a kettle full of boiling water. Guys, again, you've got to be very careful and very cautious when using hot water. Remember that dangers, accidents can always happen. You've got to be very careful. We normally will use a water bath at school. I know that you all may not have a water bath. It's too much water in there, so I'm going to drop out a bit. Okay, we just need a wee bit. We just need that. And then the test tube can jump out and rise a bit. While this incubates, I'm going to move on to the next experiment.
We looked at the test, we looked at the results from there, and we looked at the emulsification of fats. So we looked at watermarks or the oil stains there. The last test we're going to look at today is the starch test, and the reagent we're using, or the indicator we're using, is iodine. And iodine is a chemical indicator found in most labs, and again, that's the standard indicator for using when we're testing for the presence of starch. Now starch, guys, just to remind you, starch is a complex carbohydrate, and it's not like glucose, which is a simple carbohydrate monosaccharide. So hence we cannot use the same reagent that we used when testing for the presence of carbohydrates or when testing for the presence of glucose to test for the presence of starch in food samples. So what I have with me, what I have in front of me today is I've got a few different, and potatoes, and we've got some onions, and we've got apples, and again we've got some of your favorite peanut butter, and you know why it's good. We're going to test to see whether these food samples contain starch. Okay, so on this tile, this is called a spotting tile. And again, this is a very simple experiment, guys. You could do it. All you need is a dropper and a bottle of iodine, and you're going to test your different samples. Again, iodine is a bit corrosive, but as soon as you get it onto your fingers, you rinse your hands, and everything should be fine. If you do have a problem, and then it persists, then you make sure you go and see a medical specialist or somebody who can advise you accordingly. So on the spotting tile, guys, if you look very carefully, I've labeled these different wells. So these are called wells, and essentially we're going to put in the different samples into these wells, and we're going to drop in a few bits of iodine, and we're going to look for color change. Again, the first one is starch, and again, that is the positive control, and the next well is water. During the break, Indy said that somebody probably posted a question on why do we need to have a control, or what is the function of a control. Remember that having a control is important, because you know that water does not contain starch. I mean, if we were drinking water every day, we know that it would not contain starch, and hence we use that as a positive control. Often in a lab, we would use distilled water, and that is 100% pure water. It does not have any other nutrients and vitamins in it, and hence that would be an ideal control mechanism, a control test tube to use. So we use water and a positive control so that we can compare the negative test and the positive test to other samples. The question is, what if we didn't have a control? Then we would not be able to compare. And that's essentially important when you're doing any experiment, to have a control so that the experiment is fair and your results are reliable and accurate. So I'm not going to do any more talking. Let's get busy, and that's what a practical is about. So I'm going to just lift this up a bit. I'm going to elevate it a bit so that you can see exactly what we're doing, but not too much, because we're going to get a bit of these drops going through. So the first well, guys, is starch, so not too much. We're going to put in a little drop, and we're going to try and make sure that we don't contaminate the other wells, or else the results will not give us the accurate results that we need. So that's starch, a few drops in there, guys. Then we're going to look at the next one is water, and there's water here. Yeah, I'm going to try and get in some more water here, very little, not too much. Okay, whoops, just enough. Okay, and then we've got grape juice there, a bit into that, And unfortunately, I'm not going to be able to lift this up for you guys because it's going to spill, but I'm sure if we need to get closer, Wilson's going to zoom in and he's going to give you a good eye of this. Okay, so we got milk. Okay, we've got milk in here. We haven't labeled milk, but I'm going to put milk right here just for you. We've got apple. So we're going to cut a piece of apple here, guys. And I know that apple's your favorite fruit, and you know why? Because it contains lots of glucose. So we don't know if it contains starch yet, but I'm going to do an interesting test at the end, and you're going to be quite amazed with what apples contain, and actually when they're ripe, what they contain. So that's the apple that we got in there. We've also got to cut a piece of potato, and so I'm going to cut the entire one, guys, and I'm going to just take a small piece in there, and I'm going to put it in there. Okay, so that's that. Don't need a big chunk, you just need a small chunk there. We've got glucose, which we need to test, and that's glucose is, okay, grape juice, starch, glucose solution. Okay, there's it right here. Okay, getting myself confused now. That's starch in the glucose in there, and we've got a bit of eggs. We need to test the presence of starch in eggs, guys. And you can try various samples, guys. The more you try, the better your understanding of the subject would be. And you can try this at home. You can easily get iodine at a pharmacy, and you can try it out. Okay, and we've got onions, and I've brought onion intentionally because I know that Indy likes onions, and I'm not too sure she's probably going to have this at the end with the apple.
How did you know I love onions? Maybe it's because of onion breath.
And then obviously we've got some peanut butter, and that's actually Indy's favorite. Okay, so we're going to put a bit of that in there, and we're going
I
to
do love onions.
Okay.
It's actually the truth.
And
How did
actually,
you know? I was actually quite surprised.
the onions actually have a very good cooling effect on the stomach, and they also have lots of medicinal properties, and hence we have onions. And maybe it's not because it tastes good, it definitely doesn't taste as good as it does, but it obviously has very good astringent properties for the skin as well as for the stomach. So that's a very quick lesson in that. So that's what we've gotten. I'm going to very quickly put a few drops of iodine into this, and I actually want to show you, and we know that potatoes contain starch, but we got to try out in our positive control. So that's our positive control, and I'm just going to add in a drop in the positive control and the negative control. So I'm going to lift this up slightly, guys, and I'm sure you guys can see from home. Here we have starch. In the presence of starch, a substance will, if you look at this, guys, okay, I'm going to try this on here, on the potato. Okay, so if you guys can, I'm going to let it soak a bit, and then I'm going to turn it towards you so that you can clearly see. Okay, we often end up with spills, but that's fine. Here's a piece of potato that we cut, a potato that we cut through, and we can clearly see. If we look at the color of the iodine, the color of iodine initially, guys, I'm going to drop a bit of, I'm going to bring this closer towards you. Color of iodine here, guys, is normally yellowish brown. Okay, in the presence of iodine, iodine will turn a dark black or bluish precipitate, and you can see it's a dark color, and this would be the positive test. If we look at the different wells, guys, we're going to clearly see now color change, and you've got to look very quick. So let's look at in grape. In grape, no color change. Let's go to the piece of potato. So we obviously tested that, so that's a positive color. The glucose, standard glucose solution, again it remains yellowish brown, so obviously the absence of starch. Let's look at eggs, and what would you think? No starch in eggs, and we know that, guys. Here's the apple, and okay, I know you guys can't really see this. I'm going to try and lift this up as we okay, see, that's an onion, and interesting, you can see, I'm going to show you that on a bigger piece, and that's peanut butter. Okay, and here you clearly see, I'm going to try and rub this in a bit so that we have a nice color. So if you guys come, I'm going to try and move this around and come right close to the camera so that you can see this a bit better. Okay, so right here in front of me, guys, we see we've got starch, we've got potatoes, we've got apples, water, glucose, amino acids. And if you look carefully, you see the peanut butter has turned black. The onions have got some spots of black. The apple as well also contains starch. Here's the potato, which is tested positive. So of these samples, peanut butter contains starch, your onions contain starch, apples a bit of starch, potatoes definitely tested positive. Glucose does not contain starch, your eggs don't contain starch, and your grape do not contain starch. So we've looked at these here, and as I wanted to show you the experiment with the apple, I'm going to show you very quickly what happens with the apple. Okay, so if we were to cut this apple into half, guys, and clearly see this here, okay, and if we do this, and this was a question that I set in an exam, and I asked the boys, why is it that, I teach boys, and hence I use the word boys, but I ask the learners, guys, why is it that when we test for the presence of starch in apples, the color would test positive? Now, the presence of starch in apples would indicate the sweetness of the apple. So when we look at apples as they mature, apples would initially have lots of starch, and as they ripen, the starch would be converted into glucose, and that is how they taste sweet. And hence, you'll find that here we have a positive test indicating that lots of starch in apples. And as the fruit ripens, it becomes sweeter, and hence you have the sweet taste.
Let's go to a short break now. Remember, if you are struggling and you need help, remember to post your comments and questions on our Facebook page, facebook.com/LearnExtra, or on Twitter @LearnExtra. Don't go away. We'll be right back. Welcome back, guys. Let's get into some more revision. Over to you.
I'm going to click on a link very quickly for you guys to show you how big things are in relation to each other. When we look at this interactive, and you guys can find the link at the end of the notes to this. Here
I'll
we've
also
got,
put it on Facebook. I love Cells Alive.
so super, you guys can try this out. It's quite interactive, and it's about trying it out. And here we have, on a needle, we have a strand of hair. I'm going to go back, guys, to show you. Okay, so that's, okay, it's obviously not moving back there. Okay, so there's, okay, so as we move in gradually into this, we can see that's a pinhead, and as we move, there we have a hair follicle sitting on a pinhead. If we go greater into the details, we see on that we've got some microorganisms that are there, but we're going to find and look at. So that is at a magnification of almost 100 times greater in size. If we go further down and start increasing the magnification, here we see some cells. And if we talk of dust mites, here's a dust mite, and these are the dust mites that you'd find in your carpets, and sometimes they are quite big in relation to sub-microscopic organisms, and that is what you'll be able to see with a hand lens or dissecting microscope. You do not necessarily need a light microscope to see a dust mite because it's much larger. If we go further, guys, further down, we're going down. This is about 1,000 times now, so we magnify these objects 1,000 times. Here we see a red blood cell, guys. Here we see a red blood cell, and here we're going to look at, so we looked at the dust mite. Here we're seeing pollen, and this is the pollen that we have on plants, and it's from a ragweed plant, and here you can see in relation to a red blood cell, its size. We can go further down into a red blood cell, and that's a red blood cell right here, showing us its size in relation to a ragweed pollen. If we go further down into a lymphocyte, now lymphocytes are cells that we will do later on when we look at composition of blood cells. You would see that lymphocytes are much larger than red blood cells. But again, if we look at something like an E. coli, guys, and these are E. coli here. Here, if we see E. coli, guys, these are the E. coli. Now, these are bacteria that are found around us, and they play a very important role. They're found in the digestive tract, and here you can see in relation to a red blood cell, they're extremely small. And this is a scale showing us that this represents 2 micrometers in length. So if we look at a red blood cell, it's much larger than 2 micrometers in length, but if we look at a bacterial cell, in relation to that, it's almost 2 microns in length. If we go further down to looking at viruses, which are much smaller than red blood cells, here we're seeing the Ebola virus, which is this here. If you look at that, and that is 200 nanometers in length. So we can increase that. We can go further down and look at a rhinovirus, which is one of the smallest viruses that you possibly would see. And there's the magnification here. So these are extremely small. So you can play around with this, guys. It's a link to this is is attached to the notes. It's on Facebook as well. It's quite interesting to play around with these different learning tools. And remember, the more you interact with these, especially with a topic like cells, when you actually cannot see cells in life, you've got to use technology, you've got to use media to try and enhance your learning. And that's essentially what we try to encourage you. We try and give you the best opportunities, the best links to link onto and to enhance learning. Now, and I think this is important, guys, and I've always said this, create a vocabulary list of terms that you constantly refer to. So now I do this at the beginning of any chapter that I teach, and you should do this as well when you're preparing your notes. Create a list of all the terms that are important to you. And these are the terms that we're going to refer to, And during your studying, you can easily refer to them, write on what they mean, so that you can refer to them when you're in doubt or you're not too sure. I'm going to throw Knox in the deep end, and I'm going to ask him what's happening on the page, and if he can give us some feedback on the activity on the page. Knox.
Knox?
Yes, Eugene, thank you very much. We have Garabo Duffy Man Hollywood Boy Maluka. That's a very long name, by the way.
Karabo, very long.
Very long. It's Garabo Duffy Man.
Well, he hasn't got a question,
Yeah.
and there aren't any questions on the page. So what I'm telling the mindsets to do is, guys, please post your questions on the page. So next time when Knox is dropped in the hot water, he actually has a question to ask. But I said to Knox that he should read this one out anyway.
Sure. Garabo says that mindsetters, please help me grow my page. My username for the page is www.facebook.com/distinction room, just in case, please.
Yeah, so Karabo, if you missed that, guys, yesterday he was the winner of the Pay It Forward campaign, and what he did was he started this Facebook page to help other mindsetters with maths and physical science, and it's www.facebook.com/distinction room. We will reshare, and Karabo, big up to you for taking that initiative,
Sure,
right?
I think it's a great one, actually.
Yeah, cool. Thanks,
Awesome.
Eugen.
Awesome stuff. So we're going to continue, guys. So we're going to look at the microscope. Here I've got the parts of the microscope, the various parts labeled on this diagram. And what's good about having a microscope is that we can obviously use it in the lab, but today I have two microscopes that I want to show you, or two different apparatus that actually enhance the ability to look at substances or objects that are submicroscopic or extremely small that cannot be seen with the naked eye. So I'm going to very quickly look at the diagram that we have here that's labeled to show you the parts, and then we get into looking at the actual microscope that we have. So we have obviously a microscope. This is a light microscope, guys. You have an ocular piece, and that's the eyepiece. Here you have the arm that holds the parts together. We have the revolving condenser and the nose piece, which has the different objectives on them. We have the coarse adjustment, the fine adjustment. But I know that these notes are there for you, and that's the reason why I've gone through them. But I want to get over to and showing you how to use a microscope. Not necessarily how to prepare a slide today. We'll do that next week. But just to look at the parts of a microscope so that you understand the parts. And if you asked a question to label the parts and their functions, you understand exactly what these parts are. So let's go and have a look at these. Look at this. So we have two microscopes here, guys. We've got a light microscope. And I'm sure you guys know why it's called a light microscope, because obviously it requires a source of light to be able to work with. And also we have here, and this looks a bit weird, but it's a binocular, it's a dissecting microscope. And when do you use a dissecting microscope? You'll find that often you probably have a seed that you want to look at. You probably would have something that you cannot prepare a slide with. That is a little larger than something that is microscopic. So if we want to look at a hair follicle, we could look at a hair follicle here. If we want to look at a dust mite, we could see a dust mite with this. And this simply means placing a specimen onto so here I'm going to place a coin and onto the stage, and then we've got a coarse adjustment on this here, and we have a fine adjustment on the ocular pieces, which help us to look at substances or the external structure of substances. So we could look at a leaf, we could look at the stomata of the leaf, and this is quite a very good tool to look at when you're studying mosses and ferns. So you don't really need to prepare a slide. You put on the specimen and you're looking at the external view of the specimen. The light microscope, however, you'll need to prepare a slide. And today I'm not going to spend time preparing slides. I'm going to show you how to prepare a slide next week when we look at plant cells. But I thought that it's important for us to understand how to use a microscope. And very important, guys, when you have a microscope, you've got to place the microscope on a stable, flat surface. And obviously, I've got this placed at an angle so that you can see this, but it needs to be placed in such a manner that it faces you and you're behind the arm, and the coarse adjustment is closer to you. I'm going to turn this at a slight angle so that you can see when I refer to the parts, the different parts. So we call this a light microscope, obviously because there's a source of light that comes in from a lamp in here that we switch on, and that light strikes through the diaphragm. Okay, so firstly, when we look at a microscope, we look through the microscope through an eyepiece, and that's called an ocular piece. And this obviously has a lens which helps the initial magnification. So this has got a calibration of 10 times. So this will magnify whatever we're seeing at least 10 times. Then we have, this is called the nose piece, and on this nose piece we have different objectives that revolve. And the idea of having different objectives is to be able to look at your specimen under different magnifications. And always when you're preparing a slide, you got to set your microscope on a flat surface, you put your stage down, and then you place on your slide, and then you adjust your objectives to the lowest magnification possible, and then you place on your slide, and then you start observing. So we have your revolving nose piece, which have different objectives, and these basically magnify the specimens to different magnification. If we wanted to see the nucleus of a cell, we would need to use the highest magnification. If we wanted to see the cell wall, guys, you'd probably be able to see the cell wall of plant cells using the lowest magnification. So you're going to place, we're going to do the details. You place your slide on there, and you need to adjust the specimen. You have two types of adjustment. You have a coarse adjustment, guys, and a fine adjustment. And it's very important for you to understand this. When you place your slide on the stage, you need some microscopes have stage clamps that clamp the slide onto the stage. Here we've got also a similar clamp, but this clamp allows for us to move the slide so that we do not have to physically touch the slide once it's on the stage under the objectives. We also see here at the bottom, we see the diaphragm. And guys, the diaphragm is basically there to control the amount of light. And the fundamental concept behind using a microscope is using light that passes through the specimen, and that light falls on your retina, and that is the image of what you're seeing. You'll find that sometimes we do not control the light correctly, and the images that are created are not as clear as we need to. So very important is that you should be able to control the amount of light that falls on the specimen, and that is very important. So you place your slide on there, and you adjust the light, you adjust your coarse adjustment, and then we move on to the fine adjustment. The fine adjustment is used for clearing the accuracy of your image, and often you find it a bit hazy, and you can change that by playing with the fine adjustment. After that you will then change onto a higher magnification, and you probably get onto the highest objective in terms of the strongest power of the objective lens. And when you're on this objective, guys, you got to remember that at this point of time, the space between your slide and your lens is very little.
Well, guys, we've come to the end of today's show. Thanks for joining me. Remember to visit our website, learnextra.co.za/live, to get the notes and to watch the videos. And if you are stuck and you have any questions, send an email to helpdesk@learnextra.co.za and we will help you. Also, guys, we are dedicating the month of March to Library Month. Join us as we discuss some great books and some exciting competitions on the way. Goodbye.